All right, you should be able to see my screen. You also note that you can follow along with this presentation live on your own computer if Zoom is a little bit laggy for you. You can go to that link at the bottom. It'll be at the bottom of all the other slides, and it will advance the slides live while I advance them. So that might be an option if you have had trouble in the past with Zoom's own native slide thing. Sorry, I'm just making sure that my microphone is the right mic, and now it is. So this should be a lot better in quality. Okay. Hi, thank you. My name is Acacia Ackles. I'm a PhD student at Michigan State University in the Department of Integrative Biology, and I am presenting on rank epistasis, which is a new metric for measuring epistatic interactions in the absence of quantifiable fitness interactions. So I like to get my acknowledgements at the beginning of a talk rather than at the end of the talk. So I'd like to acknowledge the funding sources and the people that made this work possible, and as well as that Michigan State University, where this is conducted, is on the ancestral, traditional, and contemporary lands of the Anishinaabe Three Fires Confederacy of Ojibwe, Odawa, and Potawatomi peoples. So much like things like life or consciousness, epistasis is one of those terms that you would think has a really solid and universal definition and absolutely does not. So just to get out of the way at the very beginning, what the definition of epistasis that I'll be using is that when you think of genetics and genetic effects, we have some naive idea that they should stack on top of each other in a simple way. 1 + 1 is 2. Epistasis is the deviation from that baseline expectation. So for some example, for sort of like a diagrammatic explanation of what that means, here's some ways that fitness effects could combine with no epistasis. So A and B would be individual fitnesses, and AB is the combination of them. Here you're just looking at it in terms of, for example, like a vector addition. So they are just stacking on top of one another. In positive epistasis, you get a more beneficial effect than you would expect just from adding the mutations together. And in negative epistasis, you get a more deleterious effect than you might expect. For some quick biological examples of what this could look like, things like receptors that evolve instability, reduced generality, and unstable specificity, all of which are things that make it harder to bind to any general molecule or make it harder to survive, all put together, create this nice interlink of stable specificity. And I can talk more about that particular example later if you're interested, but just know that different things can combine in unexpected ways. And for an example of negative epistasis, you need multiple genes to lose sort of cell regulation functions in order to have cancerous cells run rampant. So who cares about this? Why are we interested? Well, because epistatic interactions form the basis of complex genetic structures. If everything interacted independently, we could really easily optimize for evolutionary fitness. So instead, if we want to understand complex structures, we need to first understand which sites or which kinds of genes are interacting epistatically. Now, people have looked at this in the past in a variety of ways. This is one of the more common equations that people have used to quantify epistasis. It's not important what exactly this is. It's just that some of the things that it involves are the number of mutations, the average fitness type of a genotype with a certain number of mutations, the effect of those mutations on their own, and the effect of those mutations combined. And it's that beta parameter, the effect of the combined mutations, that is our metric of epistasis in a system like this. Now, there are some limitations to that kind of a system, right? First of all, what number do we assign to fitness when we're thinking about the fitness of an organism with certain mutations? We can tell in certain cases that some phenotypes are better than others, but we can't necessarily assign a fitness value to that without it being sort of arbitrary. And more importantly, we don't always know what that baseline expectation is, that alpha parameter or that additive parameter, that white arrow in that diagram. We don't always know whether effects should add together just stacked on top of each other, whether they should be multiplicative effects or additive effects. And there's a lot of papers that use one expectation or the other and find very different results. So not knowing the baseline expectation, not even knowing sometimes which genes are interacting, can be a big barrier when we're trying to look at epistatic interactions. So in order to combat this sort of problem, we developed a rank-based metric of epistasis, which is inspired by non-parametric statistics. This rank-based metric doesn't require things like knowledge of exactly which sites are interacting, and it doesn't require precise fitness values to compare. So just to step you through what this model looks like and what our methods were here. First, what we do is rank all single mutant variants in a population in order of decreasing fitness. So what this means is, if this is our genome, this sort of six locus genome, we mutate each value. Here we're using a binary genome. You could use a multi-locus or multi-allele kind of genome, but that would be very hard to draw out on a slide, but it's possible. And we just get their fitnesses, and then we rank them. We sort them by which one is the most fit, with ties being averaged out. Next, we take that ranking, and what we do is we just select a target locus. In this case, it's the second one, and we mutate that site. And then we combine that with all of the other single-step mutations that we used, which creates this sort of landscape of double mutants, so that we can see then the difference between mutations on their own and mutations in combination with the target locus, which gives us a good sense of how these sites might be interacting and how important that interaction is. The way that we quantify that importance of that interaction is by we rank the sites that we rank the double mutants the same way we rank the single mutants, order them in order of fitness and create these ties, and then we compare the two lists, right? So these are the single mutants and their corresponding double mutants, sorted and compared by the initial ranking. So what we're seeing here is the difference between the initial ranking with just the single mutations and the ranking after we add that other single mutation in. And that shows us how much change has sort of been produced in the population using these mutations. I will note here, and then we run that ranking through Wilcoxon's signed rank sum variable W. We don't do the entire signed rank sum test. We just use the W that comes out of that for a variety of reasons. And I will note that the way that we do this here is a little bit different than if you go to read the extended abstract later, because this model is still in development. Some things change pretty quickly. Mostly just instead of looking population-wide, we're looking at like the entire fitness landscape because this is a digital system. And so now that we had this nice metric, we wanted to have a landscape to test it on. So what we ended up using was NK landscapes, which I'll go through pretty quickly, but N would be the number of sites, K would be the number of neighbors that they interact with, and you just go along the genome and compare all of those combinations of genes. The reason that we used this is it's a pretty well-established landscape, and it's pretty easy to see where how to increase epistasis here. Increasing the K leads to the increased interaction between sites, which is therefore increased epistasis. And the particular parameters we used, we had 200 sites and 2, 4, and 9 interacting, neighbored interacting sites. We did this in the modular agent-based evolver system MABE, which is a nice framework you can read more about. So just to recap before I start showing you results and things like that, what we're doing here is testing our rank-based epistasis metric on NK fitness landscapes to see whether it has viability as a measure of epistasis on this known landscape without assumptions of interactivity. So we don't tell the metric what sites are interacting with which other sites. So we expect to see that sites with higher degrees of epistatic activity will score higher, without any a priori assumptions about which genes are interacting with each other or how they interact. So finally, the results. Here I'll just first show you one target site for one organism across replicates. On the x-axis here, you'll see the number of sites, which is k, and on the y-axis, you'll see that Wilcoxon sign rank sum, which is the epistasis metric we're using. And you can see that that sum tracks pretty nicely with an increase in k. We see a higher metric for our epistatic measure with a higher k. If we average across all the organisms in the population, we see this very similar result. And these means here are the white dots, and these are highly statistically significant. R got pretty mad at us when we asked it to show us a value that small, so that was encouraging. And then if we want to look across the genome in sort of a slice across the entire genome, we can look averaged across all loci at sort of this nice striation of the W metrics. So at the bottom there you have K is 2, and then 4, and then 9, and you can see that the averages pretty nicely tick up with each K. So just as a wrap up, we did find that sites with higher degrees of epistatic activity scored higher across, and we had some changing NK landscape data here that I forgot to take out of this slide, but in general, higher K led to higher rank epistasis. We also expected that we would see this regardless of any a priori assumptions about which genes are interacting or how they interact, and we did. We saw this higher rank epistasis on average, even though the metric had no idea which sites were interacting with each other. You know, sites were being compared across the entire 200 site genome, when only 2 sites at a time were interacting, and it still managed to pick up on that difference. So in the future, what we would like to do with this is tune the metric for more fine-grained differences between levels of epistatic activity, compare it with other metrics to see the old information in a new light, experiment on some digital landscapes with some more complex genetic architectures, and we'd like to apply this to biological wet lab data to see what we can really get out of this really interesting new metric that we have. So that is the end of the talk. And I am happy to take any questions.
Great, thank you so much for that. Great. So yes, questions, who has one? Okay, again, I'll wait just one more couple of seconds to see if somebody who hasn't asked a question yet would like to ask one. But yeah, okay, so Hiroki, you have a question.
No, Susan was first. Go ahead, Susan.
Okay, thanks, Hiroki. I'll take advantage of your kindness. Yes, this is a very nice piece of work. Thanks for that. Very interesting. You've only done it for one size of landscape, 200, and there's a bit of noise. Do you have a feeling for how the noise will scale with increasing N? Do you expect the bounds to get a lot tighter? And kind of what size of N would you want to go for?
Yeah, that's a really good question. I don't have a sense immediately of how it might scale with increasing N. I know that increasing N tends to have a less of an effect than increasing K in these kinds of landscapes when I've used them for other projects. But that would be really interesting to look at. We definitely want to apply it to a large variety of other landscapes.
All right, and yeah, Hiroki, if you want to go ahead and ask your question, we'll take that as the final one.
Yeah, thank you very much. This is great work. So my question is how you could actually extend this methodology to be practically useful for the empirical experimental data, because you need to do a lot of fitness evaluation to calculate W, right? Mm-hmm. So do you have any idea how to sell this to experimentalists?
Yeah, that's a really good question. So experimentalists are already sort of starting to develop complete fitness landscapes. And one way that we can sell this is it actually requires much less comparison than existing measures of epistasis. So if they're already interested in looking at epistatic interactions, instead of having to test every double interaction, every triple interaction, etc., all you have to do is test sort of twice the number of sites you're looking at. So all you have to
Okay.
do is test the first, and so it actually ends up being much more computationally efficient.
Okay, that's good to know. Thank you. Perfect.
Thank you.
Okay, great. Thank you so much. And again, other questions can go on